Okay, hello everyone. Welcome to the topic about the physiology of blood. Basically, this whole topic is everything about the blood. So, about its production and its usage, its function, and all. However, you have to bear in mind that the topic of blood here is actually mainly focused on red blood cells. Not because that the other types of cells are not important. It's just because that the white blood cells sections has been mainly covered in your second year when you studied your immunology, and the other part about platelets has been covered, about the process of blood coagulation has been covered in your third year when we were talking about the anticoagulation drugs. So most of the other parts are covered in a way. So therefore, in this particular last section, we mainly cover we will talk more about red blood cells in particular. So bear in mind the overall blood. So obviously you can see after centrifugation from the blood samples, you actually have the plasma and also the red blood cells. Right, there's a little bit of buffy coat in the middle, which is composed of the white blood cells and platelets. Right. So you need to know where they are, and if it's a blood collection tube, so you need to know which type of tube they actually need. So this is something that you need to know before actually do the blood collection. So you need to know the purposes for which type of analysis that you go for. Okay, so to know whether is there any disease which are related, so in relation to blood, so obviously most of the time they actually need to do a detailed bone marrow examination, so whereby they draw some bone marrow fluid, right, from the backbone in a way. So from there they'll know whether there's anything wrong, for example, in the diagnosis of leukemia, for example, to see if there's any cancerous cells in that area or anything wrong with that. So that would be part of the diagnosis as well. So when we talk about red blood cells, as you already studied years and years ago, so it basically looks a little bit like a donut-ish. So there's no cell nucleus in a way, because you want to increase the surface area, for oxygen, for maximal oxygen carrying transport capacity, right? And when you look at it, there's 4 chains, so there's 2 alpha and 2 beta chains, so you have to bear in mind. So the alpha and beta ones are the mature ones that you can find in the adult base red blood cells, right? So this is, and there's also in the middle there's a ferrous, which is also an important part of the heme, to form the heme section. Right, if we move on to the next slide, so there's a different components of the blood as shown in the earlier one. So this is just a breakdown on the percentage of it, so you can just go through them. So you've got some proteins in the plasma. Obviously most of it is water, and you've got some solutes. So the proteins, there's a few main types, so you should roughly know what it's the function of each of them. So you've got albumin, you've got globulins, you've got fibrinogen, right? So and the red blood cells will be most of it in a way, and for the white blood cells, most of it is neutrophils and the others, and leukocytes is the lymphocytes is the next one. Sorry. Right, so when you look into the function, so it's detailed in the table over here. So you can see albumin is the most abundant one, right? So it helps to maintain osmotic pressure, and also to so obviously when you maintain osmotic pressure, so it determines where the direction of the fluid going in or out at the section. So you've got globulin, so this is large protein, so like immunoglobulin obviously helps more in the attack, because from the name is immuno, right? The attack of virus and bacteria. So you've got other globulins which are involved in the transport of other important substances and vitamins in the body. You've got fibrinogen, so from the name you already know it's platelet related, so it's more on the blood clotting side. So obviously there's other electrolytes and so on and so forth. There's enzymes, hormones, etc., in the system. Right, so what the origin of the different blood cells? So you can see the main one is the pluripotent stem cell, which has the ability to transform into different types of and transform and differentiate into different types of blood cells. As you can see here, so we've got the red blood cells one is actually on its one section on its own, and we've got platelets on its own over here, and you've got the the white blood cells one over here. So this is the main implication of this is you can see that obviously it's a multi-step process. And so for example, if a person after undergoing chemotherapy, so which type of the growth factors or stimulus that you need to provide to the individual, so it depends on what is lacking and then which part of the pathway that you want to actually activate it. Right, and again, the other implication is on your leukemia, so which type of leukemia and which type of white blood cells are actually affected. Okay, depends on which section is being affected. Okie dokie, so let's move on. So one of the main part that we're going to talk about is actually on the formation, destruction, and recycling of red blood cells. Right, so you already learned part of it, so this is more like a top-up or a revision of information. So if you look at it, so the average lifespan of red blood cells is about 3 months, so it's about 120 days. So if you look at it, so the site of erythropoiesis is actually in the bone marrow, right? But do not forget that the stimuli, which is the erythropoietin secretion, is actually from the kidney, right? So what happens is that you have a destruction process, right, which actually provides breakdown into different parts, and we try to recycle them, so and to provide into the last step of remaking the new ones again. Right, so what do we need to make? So basically ingredients in a way is one is the iron, so Fe3+. Where do we get this from? So we get it from the liver, which is the storeroom in a way for the iron. So you can see when it's transporting around in the blood vessels, it's actually bind to transferrin. But once it's kept in the liver, it's actually called ferritin. It binds to the ferritin protein. So when it gets out again from the system again, it binds again together back to transferrin to get into the site of erythropoiesis, which is in the bone marrow. So second ingredient in a way, so the ferrous here is actually to make the component, as earlier on mentioned, is for the heme. Right, so as you can see, it's from the heme and it breaks down over here. Okay, next is globin. So globin is actually the protein part of it. So basically it's amino acids. So again, when it's disrupted, so it's from here. Amino acids break down, and here it goes back into this section. Vitamin B12. So this is from the GI system, from our diet in a way. Right, so this is again for the formation of it. So basically, it's from the formation of red blood cells. Right, of course, because it's erythropoiesis. Okay, so and also there's erythropoietin, which is a stimuli. Right, so another part so once everything is formed, so it goes into the blood circulation. Another part that we haven't mentioned is actually the other part of heme, which is breakdown into biliverdin, then into bilirubin. So bilirubin is the one which I'm more familiar. So it goes through the liver section and then being secreted into the small intestine, right, from the bile duct section. So again, it comes to bilirubin. So it goes down to the poo area, right? So some parts is urobilinogen. So this is the part where it can be it's formed by the breakdown of bacteria on the bilirubin. So it can go through the kidney to get out from the urine section. Okay, so details of it, so you can actually read through the slide in a way, so it's actually the same thing, but it's easier for revision, I hope. So there's also another slide that I provided here on the flow chart, so it's basically on the same information, but we can just choose which one that you prefer to use it during your revision. Okay, so next we'll move on to the regulation of erythropoiesis process. So as you can see here, there's a stimulus. So again, erythropoiesis, the process of forming more red blood cells is triggered by reduction in the oxygen transport. So one of the stimuli is probably if you go for hiking to the highest mountain in the world, you know, that kind of thing, Himalaya, or you go to hike on our Kota Kinabalu-ish. So you can actually, so the body detects there's some reduction in the oxygen level. So the receptor, so it's basically at the kidney cells. So therefore they will secrete more erythropoietin. So therefore you form more red blood cells. Once it's done, so the process will sort of slow down or stop for the moment. Okay. So this is quite straightforward. Okay, so next, so this is about the blood group. So I think this is something that you learn in Form 5, if I'm not wrong. So again, it's very simple. You've got AB, and O. So if it's A, so obviously on top of the red blood cells, you sort of have the antigen sticking out of it, which is the A. So our body sort of realized that, hello, A is our friend for antigen. So therefore, it can accept the blood A and O, but it doesn't recognize B, so please do not put B in the system. Right, therefore it's incompatible. Similarly for other blood type like B, so B has the antigen B on top of it, so that's why I can accept B blood and O blood, but I don't want anything to do with A. AB has both the A and B antigen, so anything is okay to them because they're fine. O has none of all this, so basically I do not recognize, the body does not recognize the antigen A or B, so everything is foreign, so we do not like them, so it's incompatible. Right, so next, moving on to something that you already learned before, is about hemolytic disease of the newborn. So it's basically about the rhesus. So this only occurs when the mom is a rhesus negative, but the baby is a rhesus positive. So you can assume that the positive comes from the papa side, right? So the genetic from the dad side. So from the negative, so what happens is that the blood circulation obviously is an independent system, right? Because it's not directly connected in a way, right? During the pregnancy, right? But there's obviously diffusion of oxygen and so on, and ways to get out of the body and things like that. But it's a diffusion process. So what happens at the first pregnancy, there's no contact in a way until it's during delivery. So obviously when things start to break up and things like that, so things get a little bit messy during delivery. So the body get exposed to it. So that got exposed to the rhesus positive ones. So therefore, our body the mom's body produce the anti-antigen in a way that, hello, I don't like your rhesus positive. I don't know you. You look foreign to me. So it start to attack it. So that's why it's okay during the first pregnancy, right? However, it becomes an issue in the second pregnancy. So the body sort of recognize it, so therefore it can attack, so therefore they can cause an hemolytic disease in the newborn. Okay, right, details you can read through more in detail. Sorry, I accidentally flipped through it. So there are some checkpoint questions, so therefore that's all for this short class in a way. So there's some checkpoint questions from different books, so you can use as a revision guideline to know what you miss or what you need to pay attention to. So again, if it's the white blood cell related ones, you can don't focus too much for the purpose of exams, okay? But again, they are all information which are important, right? Okay, that's it for now. Thank you for your attention. See you in class. Bye-bye.